Hi, thanks for coming to visit us. My name's Gordon Wood. I'm CEO of OviGenex. OviGenex is a California-based producer of ovine collagen, including functionalized collagens. Based in Southern California, OviGenex solely sources its biomaterials from safe, industry-compliant suppliers in Western Australia. But why ovine? And why source only from Australia? The Australian Ministry of Health and the USDA both certify that Australian sheep are prion-free. This means that there is no possibility of TSCs being transmitted between the sheep and the human via zoonosis. This is clearly not the case in bovine collagen, where BSE is known to cause Creutzfeldt-Jakob disease, otherwise known as mad cow disease. Similarly, fowl collagen is associated with avian flu and not genetically close to mammals. Unlike porcine collagen, ovine products are not associated with any social or religious connotations and can be used throughout the world. Indeed, our ovine collagen products carry certified halal status. Rat tail collagens are known to carry diseases such as murine typhus and rat bite fever, whilst fish collagen is prone to heavy metal contamination and is not genetically close to humans. OviGenex uses its proprietary technology to process, purify, and functionalize ovine collagen for a variety of uses, including research, medical, veterinary, and cosmetic applications. Importantly, using our proprietary manufacturing processes, there are no detectable levels of endotoxins in our research-grade products. Functionalizing the collagen lets OviGenex add significant value and increase the efficacy of collagen materials by adding ligands and bioactive molecules to the collagen. CollOvine is equally as attractive to the cosmetic and cosmeceutical industry, as by removing the endotoxins, we not only provide a purer biomaterial, we also remove the painful sting associated with cosmeceuticals, which is caused by high levels of endotoxins. Collagen is part of the extracellular matrix that provides structural support and a healthy environment for cells. Our research-grade CollOvine can commonly be used in tissue engineering, regenerative medicine, and cancer research. Here to tell you a bit more is Dr. Bill Ernst.
Our ovine collagen, CollOvine, is subjected to a number of analytical assays. One assay that we use to measure the level of denaturation of the purified collagen. Since the purification process is extensive and it usually requires various acids and bases, and some manufacturers even use chlorine bleach, it is important to ensure that the collagen has not been denatured during the purification process. The assay shown here is a trypsin resistance assay. Since native collagen is resistant to the enzymatic activity of trypsin, we can incubate a sample of the collagen in the presence of trypsin and then analyze the sample by electrophoresis on a polyacrylamide gel. The gel is then stained with a blue dye that binds to proteins so that we can visualize the collagen. If the collagen is in its native form and has not been denatured, the collagen will separate into a characteristic banding pattern. If the sample is denatured, the collagen will be digested into small pieces and be visualized as a smear. As demonstrated in this figure, the CollOvine product shows the characteristic banding pattern in both the trypsin treated and the collagen without the trypsin, indicating that the CollOvine is resistant to trypsin and therefore is in its native state. In contrast, several competitors' collagen products from bovine or rat have been extensively digested by the trypsin, indicating that these products are denatured. It should also be noted that the collagen without the trypsin treatment is separated. When separated on the gel, even denatured collagen will show the characteristic banding pattern. Without analyzing the collagen appropriately, one will not know if the collagen is in its native form or whether it is in fact denatured. The CollOvine product is always in its native form.
Hi, I'm Gary Fuji, the Chief Scientific Officer for OviGenex, and wanted to talk a little bit about some of the research applications that we see for CollOvine products. Collagen, as you know, is part of an extracellular matrix that provides basically structural support and a very healthy environment for cells to grow. And so collagen finds quite a bit of use in cell culture for cancer research and regenerative medicine, as well as in tissue engineering. Here's for some applications that our customers have been working on. Here's a sample of an artificial bone marrow that is to be used for tissue engineering, and in particular, this is to be used as a model for cancer metastasis into bone and into bone marrow. We also have some data that from a collaborator that shows that the collagen is very native type structure with the triple helix. The HTA1080 cell line excretes an enzyme which actually degrades the collagen, and so when you put the cells on top of the collagen-coated wells that have a permeable membrane, the cells eventually migrate and invade the outside of the well. And so the data here shows that the cells do invade and get through the collagen, whereas negative control cells that don't secrete the enzyme that degrades the collagen aren't able to traverse the well properly.
Collagen is often used in cell culture as a substrate for cell growth. In this experiment, BT549 cells were used for a three-dimensional invasion assay. Here, the cells were grown into a tumorosphere and then seeded into wells for a metastatic cancer invasive study. The cells were grown in the presence of CollOvine or rat-tail collagen at 2 mg per ml, and varying the dose of the CollOvine. And here you can see that the BT549 cells could invade the surrounding area quite well over time, even at concentrations at 1 mg per ml. They were compared with various vendors and shown that the CollOvine worked just as well or better than any of the other vendors.
Hi, I'm Sam, and I'm here to introduce you to a line of functionalized CollOvine products. These products are designed to expand the possibilities of how you can use collagen in your research. For example, if you are able to conjugate a ligand to the collagen, not only will that collagen act as a matrix for your cells, it will also now act as a targeting agent, pulling down your cells of interest. For example, a particular cancer cell or a particular type of stem cell. To simplify the conjugation, we have designed two functionalized CollOvine listed in orange below, CollOvine PEG2-MAL and CollOvine SPDP, both of which are designed to react with a furyl sulfhydryl group, which can be easily found as a cysteine group on peptides or proteins. And it's a very common conjugation chemistry. The third functionalized CollOvine product you can find on our website today, listed below in orange, is CollOvine MA, CollOvine methacrylate. This particular functionalized collagen is not for the conjugation to a ligand, but it is a cross-linkable collagen that can form stable 3D hydrogel matrices for different purposes. I'm going to go over how you can use this functionalized CollOvine in the next few slides. If you do not find the specific chemistry for your need, we have a great team of scientists, including myself, that can help you design a specific functionalized CollOvine for your need. You can contact us for further details. But now I'm going to go through how you can use these three specific collagen derivatives in the next few slides. In this slide, I will show you how you can conjugate your ligand of interest to the CollOvine PEG2-MAL. Shown below is a schematic drawing of CollOvine PEG2-MAL as molecule A. All you have to do is to add your ligand of interest with a free sulfhydryl group indicated as SH here in molecule B. In our example, we're using a recombinant aspergillus protein, RSBF9, for conjugation. We picked this protein because it contains a cysteine group as an amino acid in the protein for conjugation. When you mix A and B together in the correct buffer, it will easily form the conjugate C. To confirm that we have conjugation, we've ran a protein gel. On the left panel with blue hue is a Kumasi stained protein gel, and lane A is loaded with the CollOvine PEG2-MAL alone. You will see a major band that's greater than 200 kilodaltons. And lane B is loaded with the RSBF9 our model protein alone. And in lane C is the conjugate reaction. You can still see some free RSBF9 protein left in lane C because we've added an excess amount of RSBF9 compared to CollOvine PEG2-MAL to ensure conjugation. The conjugation success is indicated by the shift of the band in molecular weight from lane A to lane C as shown in my red arrow. It's kind of hard to see the shift in this picture because the molecular weight is very high, but we have further confirmed that the conjugation happened by running a Western blot, shown in the gray-hued panel on the right. Again, in lane A is loaded with CollOvine PEG2-MAL alone. There's no dark band because we reacted the gel with an antibody that will only react with the RSBF9 protein. So there's no dark band in lane A. But in lane B, because it's loaded with RSBF9 alone, there's several bands that has reacted with the antibody. In lane C, not only does the same bands in lane B is shown where the free RSBF9 is, there is an extra band that's not present in lane A nor in lane B with high molecular weight that reacts with the RSBF9 antibody, indicating that that's where the collagen RSBF9 conjugate is. So I've shown you here that a protein containing a sulfhydryl group can react to the CollOvine PEG2-MAL. So we've done something similar with the CollOvine SPDP with our model protein as a conjugation example. Shown below is the CollOvine SPDP schematic drawing as molecule A. Again, we've added model protein, the recombinant aspergillus protein RSBF9 as molecule B, and it contains the cysteine amino acid for conjugation. After we have separately reduced molecule A and molecule B, we've mixed the reduced molecules together, and in the right buffer, conjugate C is formed. To confirm that we have the conjugate, Again, we've run a protein gel and a Western blot. On the left panel with the blue hue is a Comasi stain protein gel of the 3 molecules. Lane A is loaded with CollOvine SPDP alone. Lane B is loaded with RS-BEF9 alone. And lane C is the conjugation reaction of A and B. And you can see that in lane A, the 3-4 major bands has disappeared into sort of a smear in lane C, which indicates that the conjugation has formed, but there is, each CollOvine SPDP has different amounts of RS-BEF9 conjugated to it. This is confirmed by the Western blot as shown on the right-hand panel with the black and white hue. We've run the Western using the RS-BEF9 antibody again. So in lane A, because it's only loaded with CollOvine SPDP, it did not react with the antibody. In lane B, is only loaded with the RS-BEF9 protein, and so it has the bands that's reacted with the RS-BEF9 antibody. In lane C, you see the smear again that's not present in either lane A or lane B, which indicates that that's where the conjugate is and that each of the CollOvine SPDP has different amount of RS-BEF9 conjugated to it. So we've shown here that you can conjugate a protein with a sulfhydryl group to CollOvine SPDP. So now I'm going to show you how you can use these CollOvine conjugates in real life for your research. In this example, I've conjugated a recombinant integrin binding protein to one of the functionalized CollOvine. As some of you already know, a lot of the cancer cell lines are known to overly express integrins on its cell surfaces. So in this example, I'm going to show you how we can use these CollOvine RIBP conjugates to pull down these integrin expressing cells. So I coated 2 wells on a tissue culture plate. In one well is coated with CollOvine alone, and in another well is coated with the CollOvine RIBP conjugate. I then added the same amount of integrin expressing cells to each of the wells, and after a short incubation time, I rinsed off the cells that did not bind to the well surfaces. I let it grow for at least 24 hours, and at the end of the incubation period, I measured the amount of cells that are growing on the plates using a fluorescent indicator, for example, Alamo blue. So as you can see in the bar graph below, the well that's coated with the CollOvine RIBP conjugate has about 50% higher fluorescence than the well that is coated with CollOvine alone, indicated that there's a lot more cells growing in the CollOvine RIBP conjugate coated wells than the CollOvine wells alone. So this is a very simple example of how you can use CollOvine conjugates to pull down cells of interest. And I'm sure as I was speaking, you already have a lot of ideas of how you can use these CollOvine conjugates in your research. And we'll be happy to hear what your ideas are, what you would like to do, and feel free to reach out to us to try out one of these functionalized CollOvine, and we'll be happy to design an experiment with you and also help you customize the conjugation chemistry and even help you with the conjugation of your ligands to the collagen. So feel free to reach out. The last functionalized CollOvine that I want to cover today is CollOvine methacrylate, CollOvine MA. The chemistry of how we form this CollOvine MA is shown in the schematic below. These CollOvine MAs are provided to customers who want to form hydrogels with collagen. With the help of a photoinitiator, which we can provide to you, these hydrogels can cross-link and form within 10 minutes with UV exposure. An example of how these hydrogels formed under these conditions will look like is shown in the picture below. You can use these hydrogels in your typical hydrogel applications, such as trapping cells and growing them in these 3D matrices. Again, this CollOvine MA is available on the website today. Feel free to reach out for a sample for your next hydrogel experiment.
In conclusion, collagen is an important tool in various research areas. CollOvine provides comparable or better results when compared to collagen from other species and vendors. Other applications of CollOvine include drug delivery, stem cell research, and assay development. With that, I'd like to hand you over to Dr. Stuart White. Stuart heads up our European operations, and he's happy to take any questions you may have. We look forward to seeing you in person next time. Enjoy the rest of Congress.